Welcome to what I'm hoping will be a relatively short lesson on feedback and homeostasis. As you can see, a wonderful example from the lazier of my cats. So, what is homeostasis? I'm hoping that after all these years of science classes, you do know what that means. It's basically the active maintenance of stable internal conditions. The fact that it's active means that it requires energy. So it is something that your body or your cells are actively maintaining, and they're keeping conditions the same inside. This happens at multiple levels of organization, anywhere from osmoregulation within a single cell to maintain your solute concentrations and to keep the water potential at the right levels, to maintaining the proper amounts of blood sugar in an organism so that way their cells and organs are able to function. Feedback is when you modify or control something based on the results or its effects. So we're going to look at feedback loops and we're going to look at homeostasis today. So first we're going to look at negative feedback. Negative feedback loops are designed to maintain homeostasis and they correct any imbalances. So what happens is there will be some sort of condition. Let's say we're looking at levels of some molecule in the blood. And so this optimal condition is known as the set point. And it varies from thing to thing. So you'll have an optimal condition or set point for blood sugar, for blood calcium, for solute concentrations, for blood pressure. All of these things have set points. Then what happens is if anything deviates from these set points, you have sensors in whatever part of the body or cell that detect any deviations. So for example, for calcium levels, you have calcium receptors in, I believe it's your hypothalamus, that will detect how much calcium is in the blood. If the levels are too low, then what they'll do is they'll send a message to a control center. The control center in a lot of the endocrine cases happens to be the pituitary gland, which mediates a lot of hormone release, but it can be anything depending on whether we're looking at the organism level or whether we're looking at the cellular level. And then finally, an effector, something that has an effect, causes a response, and that response restores conditions back to the original. So the entire point of this is to get back to your original conditions. So let's look at a few examples. We've actually already seen one of these examples when we were looking at metabolic pathways. What we have here is we have threonine. It is the initial molecule in a metabolic pathway by which a number of enzymes, here 5 different enzymes, are basically transforming threonine into different forms until finally you get the product isoleucine. Now, in order to make sure that you don't have the cell, wasting a lot of energy, making a bunch of unneeded isoleucine, what happens is isoleucine ends up acting as an inhibitor of that first enzyme in the pathway. That way, once you get enough isoleucine, you stop making isoleucine. If the amount of isoleucine decreases, then that blocks the inhibition and or stops the inhibition, and then enzyme 1 is able to function again. Here's an example at the organism level, and it should be a super familiar example. It is temperature regulation. So for humans, our set point is 37 degrees Celsius, otherwise known as 98.6 degrees Fahrenheit. Our control center consists of temperature sensors in the hypothalamus. So if they sense a rise in body temperature, they'll initiate a bunch of responses throughout the body. One of those is your blood vessels dilate, they get larger, and that diverts more blood to the surface of your body, where the heat in your blood can be released into the environment. You also have activity of sweat glands. They are releasing sweat as the sweat evaporates, as the water evaporates. That removes heat from your body. That ends up, the net effect of that is to cool you down, which will hopefully return you back to the normal body temperature. There's also another feedback loop for when the opposite happens. So if your body temperature falls, that is going to make your blood vessels constrict. They're going to get narrower. That's going to divert blood away from your skin and more into your core organs. So that way you're not losing heat to the environment. It's also why if you've been outside in the cold a lot, your skin feels cold. Like if somebody else comes up and touches you, if you measured skin temperature, your skin is much colder when you've been outside in cold weather. Your sweat glands are going to be deactivated. You don't want to have any sweating. And then you have shivering activated, which are basically these contractions of muscles. It's involuntary, but it generates heat. That heat will hopefully help raise your body temperature back up to the normal. So you have a number of effectors. You have epithelial cells and blood vessels that are going to control, sorry, I made a mistake when I wrote this. That's actually supposed to be smooth muscle. Smooth muscle cells in blood vessels that will constrict or dilate to close and open the blood vessels. You have your sweat glands, and then you also have your muscles are effectors. So let's look at a less familiar example. We're going to look at abscisic acid. It's abbreviated ABA on this slide. Abscisic acid is a plant hormone, and it mediates response to environmental stressors. And the one we're going to look at is water stress. So if you have a plant and it is in a very dry environment, let's say it's experiencing a drought, the water potential is going to change. Basically, the cells will become more hypertonic. They will not be turgid. I'm bringing up some terms from our water potential notes. So cells become less turgid. Basically, they start wilting. So instead of having this nice fat cell, I'm going to try to draw the membrane inside. And there's our central vacuole. Instead of having this nice fat cell, you start having a much smaller cell, and that's bad because plants rely on water potential to help them move both water and solutes around in their body. So what happens is when the water potential or solute concentration reaches a certain point, plants will release this abscisic acid. Abscisic acid has a we're going to look at the impact it has on stomata. So what happens is it closes stomata, which are pores in a leaf, and this is a single stoma. And what abscisic acid does is it causes those to close. When they close, you are no longer releasing water into the surrounding atmosphere, and then you're able to hopefully maintain lower solute concentrations, higher amounts of water. There's also transcription activity based on this pathway, but we're mostly focusing on how it's involved in negative feedback. So let's move on. Let's look at positive feedback loops. So positive feedback loops are not about maintaining homeostasis. They're the opposite. They're trying to get you farther away from your initial condition. So you have a stimulus, that's a change in your body condition, and the response is to increase that change. It's an amplification process, and usually you want to create some sort of change. And if you're confused, let's look at a few examples. So the big example is childbirth. So when a child grows large enough and when it's basically in the right position, the head of the baby will start to press on the cervix, which is the opening to the uterus. When that happens, that's going to trigger a response where contractions begin. That puts more pressure on the cervix, which causes the release of a hormone, oxytocin. And oxytocin will basically cause the uterus to contract harder. That puts more pressure on the cervix, more oxytocin is released, that gives you more contractions until ultimately you have a change in the system and the baby is born. So here is what that looks like in diagram form. So the head of the baby, like I said, pushes against the cervix. This is the cervix right here, the opening to the vaginal canal. Then there are sensory nerves, stretch receptors in the cervix and the uterus that can send messages to the brain. When the brain receives those messages, the pituitary gland is going to release a hormone called oxytocin. Oxytocin goes through the blood and it's carried to the uterus, where it stimulates further contraction, which pushes the baby towards the cervix, which causes another nerve signal to get sent to the brain, which releases more oxytocin, which makes the contractions even stronger, until eventually the baby is born. And this makes sense. You don't want to maintain the homeostasis internal condition of being pregnant once the baby is at term. You want the baby to be born so that, it can start developing on its own, and so that way the health of the mother is preserved. So just another diagram showing you that, what's happening, and, let's look at another example. So lactation in mammals is another example of this. So what happens is two different things. When an infant suckles at its mother's breast, that's going to send sensory impulses. It's going to trigger sensory nerves, which are going to send signals to the brain, and they're going to cause the release of oxytocin. So oxytocin is sometimes called the maternal or the mother molecule because it's involved a lot in pregnancy as well as lactation. So oxytocin is released. Oxytocin is going to stimulate lactation, so the lactiferous ducts, which make milk, are going to start producing more milk, which is going to get transported down all of these tubules until it reaches the nipple, where it is able to be discharged. Another thing is these sensory nerves are going to also trigger lactocytes, which produce milk, to produce more milk, so that way it can be basically released. That increases milk production, which increases the suckling action of the infant, which then will increase milk production, and this continues until the infant is basically full. They stop suckling, and then the milk production slows down. All right. Now, I like to show you a plant example as well as an animal example. So here's one of our plant examples. Let's look at the ripening of fruit. So we talked a little in our previous lesson about ethylene, which is a gaseous hormone, and I said that it causes fruit to ripen. This is a positive feedback loop. So you don't want, if you have one fruit start ripening, you don't want it to unripen. The plant wants its fruit to ripen, so that way it can fall off of the tree, or it can fall off of the plant, and those seeds can get dispersed. In the case of fruits are just there as a bribe to tempt animals to come along and eat them, and then spread their seeds of the plant farther away from the tree. So you want ripe plants, or ripe fruits, that way animals will come eat them and spread your seeds. So a single apple on a tree ripens. It produces ethylene. That ethylene acts on neighboring fruits to cause them to ripen, and they're going to produce more ethylene, and then that is going to cause more apples to ripen, and then they're going to produce more ethylene, and this happens until eventually all of the apples on the tree are ripe. This is why, for example, you can actually, if you're trying to get an avocado to ripen, you can put it in a paper bag with ripe banana, and the banana produces ethylene. That ethylene helps get that avocado to ripen. So nice food hack if you need it. So let's look at this diagram over here on the left. So we have ethylene produced in one fruit. That gas is going to diffuse to neighboring fruits. It's going to get into their cells and interact with an ethylene receptor and cause that fruit to ripen. As a part of that ripening process, that fruit releases ethylene. That ethylene will then ripen more fruit, and as more fruit ripens, that ethylene is then going to go on to trigger more and more fruit ripening. Another example of positive feedback, you want to have blood clotting reach a certain goal. If you've got a cut in your blood vessels, you don't want that to stay in a constant condition. You want to block off that cut so that way your body doesn't lose too much blood. So what ends up happening is when you cut some of your tissue, cut some of your skin, that wounded tissue, those wounded cells, dead cells, dying cells, release chemicals, and those chemicals are going to recruit platelets in the blood. So those platelets are going to then start adhering to wherever you have the wound, and they're going to release chemicals that attract more platelets. And so more platelets come, they attract more platelets until eventually the wound clots, and at that point, the signaling stops. So this is showing you in diagram form how that happens. So you have some sort of tear in the wall of a blood vessel that starts this feedback cycle. Platelets are going to start to adhere to this break, so this area right here, and as they adhere, they're going to release chemicals. Those released chemicals attract more platelets, so more platelets adhere and release more chemicals and attract more platelets until finally you end the feedback cycle when there is no more blood leaking out of that vessel. So just like with signal transduction, when you have alterations in feedback loops, that can cause negative consequences to health. We're going to bring it back to diabetes real quick. So a healthy individual has a pancreas producing insulin. That insulin interacts with cells and causes those cells to take up glucose. Type 1 diabetes, you don't have that insulin being produced, and since you don't have that insulin being produced, your cells can't respond to take up glucose. And then in type 2 diabetes, your pancreas can produce insulin, but your insulin receptors are not receptive. This is part of the blood sugar feedback loop, which involves two hormones called insulin and glucagon. When your blood sugar is too high, then insulin is going to be released by your pancreas. If your blood sugar goes too low, then glucagon is going to be released by your pancreas, and glucagon will trigger your liver to start breaking down glycogen into glucose molecules and release those into the blood, and a number of other actions to increase your blood sugar. So when that feedback loop is interrupted, that's when you get problems like diabetes. There are also a couple of conditions, one of which is diabetes insipidus. So diabetes doesn't just refer to traditional diabetes that deals with insulin. Diabetes insipidus involves lowered amounts of antidiuretic hormone. A diuretic, is something that makes you pee. Antidiuretic prevents urination. So, that should make sense with the fact that an antidiuretic hormone signals the kidneys to keep more water, retain water, and this will happen if, let's say, you're sweating a lot, you're dehydrated, then the response to that is your hypothalamus will release this ADH. ADH will go through the bloodstream, and once it reaches the kidneys, it's going to direct them to start basically reabsorbing more water, so your urine becomes more concentrated, more water is retained, so hopefully that will basically increase your blood pressure, and then that will turn off the receptors in the hypothalamus that are triggering the release of ADH. There are some cases, like I said, diabetes insipidus and other conditions where you have lower ADH levels, so you don't release as much of this antidiuretic hormone in response to dehydration. And what that does is that allows your kidneys to release watery urine, which means you're going to lose more water. So anybody who has these conditions where they have low ADH levels has to drink a lot more water. They'll have to urinate more frequently. One of the symptoms is their urine tends to be very clear because it's very dilute. So if they have, diabetes insipidus and they don't take medication, they'll need to make sure that they're well hydrated. All right, I hope that wasn't too taxing, and the cell cycle video will hopefully be up in a couple of days.